This video is for Unit 3.4 and it's about osmosis. Osmosis is one of the types of passive transport that was covered in the video for 3.3, but I want to go into it a little more detail. This is especially important for your egg lab report. So there are three different osmotic conditions that can occur, and remember, osmosis is the movement of water to an area of high concentration of water to an area of low concentration of water. So we're going to look at how those different areas of concentration can affect the movement of water in specific situations. And these specific situations are these osmotic conditions. So the first osmotic condition is called isotonic, and an isotonic condition or environment occurs when the concentration of the solute inside the cell is the same as the concentration of the solute outside the cell. So remember back in Unit 3.3 video, I talked about the solute being the stuff that was dissolved in the water, like the Kool-Aid powder that's dissolved in the water. And so what we're really looking at here is how much solids are dissolved inside the cell and outside the cell. And then based on that, what's going to happen with the water. So in all these cases, we're going to assume our solid is glucose. And I'm going to draw glucose molecules as just little dots around the cell and in the cell. And the reason I chose glucose is because glucose is too big to go through the phospholipids of the cell membrane. So it's not going to move in and out of the cell in this case. So outside of the cell, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 glucose molecules. Now, if the concentration of the solute or the glucose outside the cell is the same as that inside the cell, I need 10 glucose molecules outside this or inside the cell as well. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. Remember that this isn't just glucose around the cell. The cell also has lots of water. So we're also going to show the water molecules. I'll just put 5 of those out there. 1, 2, 3, 4, 5. And again, if the concentration is the same, that means we have to have the same amount of water in the cell as well. Okay, so the glucose and the water are in equal amounts. So that means that there's not going to be any net movement of water. Well, what does that mean? That means these water molecules may move out through the membrane, but for every molecule that moves out, some molecules will also move in, just because of their random motion moving in and out of the cell. So the water is going to move in and out, but it's not going to build up inside the cell, and it's not going to build up outside the cell. Equal amounts will move in and out. Therefore, no net movement. The water is not going to build up in one place or the other. So again, that's called an isotonic environment for the cell. Okay, the second osmotic condition that can exist is if a cell is hypertonic. Now, if something is hyper, that means it's high. So if someone is really hyper, they have a lot of energy. So when we're talking about something being hypertonic, that means that thing, in this case the cell, has a lot of solute in it compared to the solute around it. So this time I'm going to put those 10 glucose molecules inside the cell. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And that is going to be the high area of concentration. So I'm only going to put 5 glucose molecules outside the cell. Now the water could be the same. 1, 2, 3, 4, 5, inside and out. So we're going to put 5 molecules of water inside the cell, 5 molecules outside the cell. But the concentration of glucose, the blue dots, is higher in the cell, right? There are 10 of those in the cell compared to 5 outside. So in order to even out the concentration, because right now the inside of the cell is highly concentrated with glucose, the best way to do that, since the glucose can't move, is to move water in. So the water is going to come in and essentially dilute the glucose in the cell. Again, you could think about this as like Kool-Aid. So if the Kool-Aid inside the cell is really concentrated, that means it's a really dark red Kool-Aid. The Kool-Aid outside the cell is more diluted, so it's a really light colored Kool-Aid. We want to make them equal in concentration, so we move some of the lighter colored Kool-Aid from the outside into the middle of the cell, and that makes that middle of the cell less concentrated or lighter in color. And the water will keep moving into the cell until they're equal. And we call that point the equilibrium. So at equilibrium, the water keeps moving. It moves back and forth, but it does not get to that point until the water starts moving in. So in a hypertonic cell, if the cell is hypertonic, the water moves in. All right. The next type of osmotic condition is called hypotonic. And again, the prefixes give you a clue. The prefix hypo means low. You've maybe heard the word hypothermia, when you get too cold, when your temperature gets too low. So it's the same idea in osmosis. Hypotonic refers to a low concentration of solute in the cell. So as before we had more glucose in the cell, this time we're going to have less glucose in the cell, put 5 molecules in the cell, and more on the outside. So I'll put 10 around the outside. So now the cell is hypotonic. Just going to add the water to show that that's there. Remember, the water molecules can be even, but if the glucose is more concentrated inside, the glucose is more concentrated outside, that means it's hypotonic inside the cell. So what's going to happen in this environment? Well, again, the water is going to try to even it out. So it's more concentrated on the outside, it's diluted on the inside, which means the water inside the cell is going to move out to dilute the water on the outside of the cell. So again, the water moves out. And it will continue to move out until, again, it reaches that equilibrium. And much time it will move in and out of the cell equally, but nothing will happen to the cell itself. It's not going to increase or decrease in water. Okay, so the 3 osmotic conditions were isotonic and hypertonic and hypotonic. And in each of the examples I gave, I talked about a cell as being hypertonic or hypotonic, but really it's 2 parts in each case. So when we have an example, we just think about like a tank of water with a membrane between the 2 halves. And if one side has a lot of glucose in it and the other side does not, the side that is the most concentrated is the side that is hypertonic, and the side that is less concentrated is the side that is hypotonic. So even when we're talking about the cells, if the cell is hypertonic, then the outside is hypotonic. And vice versa. If the cell is hypotonic, the outside is hypertonic. And in this case, as it says up above here, no matter what, water will always travel from hypotonic to hypertonic. So in this case, the water will move to the left. If I set it up the other way, where we had, again, our chamber, but on this side we put more glucose molecules. Remember, glucose can't move. It can't diffuse from one side to the other. It's too big. So the water is going to move instead to even things out. The left side is more dilute. It's like the pale colored Kool-Aid, so it's hypotonic. The right side is more concentrated. It's like the really dark Kool-Aid, so it's hypertonic. In order to even them out, the water has to move from hypotonic, where it's diluted, to hypertonic, where it's concentrated. Now, what does this mean for cells? Well, when water moves in or out of cells, in plant cells, if they lose water, their membrane can actually shrink. So plasmolysis is the shrinking of a plant cell's membrane because they lose water. Now let's think about why that might happen. Here's our plant cell. The water is going out of it. So what would that mean? That would mean there were more molecules of solute outside of the cell and less molecules of solute inside. That would mean that this is a hypotonic cell. So the cell itself has less molecules of solute in it, so water is going to leave the cell to fill up the environment around it. And this is what happens when a plant isn't watered for a long period of time. So there is more water in the cells than around the cells. And so water will leave those cells, shrinking those cells and causing the plant to wilt. Another effect that is called cytolysis, and this happens more in an animal cell's membrane because plant cells have cell walls that protect them a little bit more. But cytolysis is when an animal cell's membrane bursts or breaks open. Cyto means cell, lysis means to cut or to split. So we're bursting an animal cell's membrane. So why is that happening? Well, let's look again at the environment that would cause this. So if we have a cell that's membrane is bursting, that means that it's getting filled with water. Why would a cell get filled with water? Well, if there was more stuff, more solute, dissolved in the cell than outside the cell, water would move in because the cell would be hypertonic. So if your cells become hypertonic, they will start to take on water, and that creates a bursting of the cell membranes. Now, I always get the question of, is it possible for a person to drink so much water that their cells burst? And that is possible. There are some individual examples of where people drink too much water over too short a period of time, and your brain cells especially are kinds of sensitive to this, and those cells take on that water and eventually could burst. So that is a danger, not probably anything that anybody would normally deal with, but in extreme circumstances that could happen. But this diagram down here kind of shows you the progression of what would happen here. So normal blood cells are right here in the middle. And if the cell is in a hypertonic environment and it loses water, it would look like this one over here, crenellated. I don't need you to know about that one though. I'm more interested in the cytolysis. As you add more water to the environment, it's going to start going into the cell and get more swollen and until it eventually bursts, just like a balloon that's overfilled with air. So just to review, the three osmotic conditions are isotonic, hypertonic, and hypotonic. It involves the movement of water in or out of the cell from an area of high concentration to low concentration. I want you to think about these lastly now in terms of the egg lab. So when you added the egg to corn syrup, the mass of the egg shrank, which means something was coming out of the egg. When you added the egg to water, the mass of the egg increased, which means something was coming into the egg. So just like I showed up here, when this cell gained water, it was a hypertonic cell. So when you put your egg into water, the cell was hypertonic, which means it brought in more water. It gained mass. When you put your cell into corn syrup, the cell was hypotonic, which meant that it lost water and therefore lost mass. And then you need to decide what happened to your cell when you put it in your third liquid. Was it hypertonic or hypotonic when you put it in that liquid? Eventually they even out and they become isotonic when they reach that equilibrium. But when you first put that egg in the corn syrup or the water or your liquid, it's either hypo or hypertonic, and that's your job to talk about in the conclusion of your lab report.